Hey there, biology. So today we're going to go ahead and talk about RNA and protein synthesis. So this is going to cover chapter 13.1 and 13.2. What you should be able to do during this time is fill out the questions 1 through 4 in your chapter 13 study guide. And I'll also tell you to do something in your notes at the end of the video. All right, so let's go ahead and start chapter 13. Okay, so as we said, this is going to cover RNA and protein synthesis. Now, RNA actually gets a bad rap. We typically hear about DNA of the nucleic acids, but RNA is just as important because we need to get our message from our DNA out to the rest of the cells, and RNA is what makes that possible. So RNA is the other nucleic acid, and it's also made up of nucleotides. So remember, our nucleotides have a sugar, they have a phosphate group, and a nitrogen base. Now the sugar in RNA is ribose instead of deoxyribose like DNA. So ribose gives us our RNA there, where deoxyribose gives us that DNA. RNA is single-stranded. DNA, if you remember, is double-stranded. And instead of containing thymine that attaches with adenine, it's going to contain uracil. Okay, so these are big differences, and you should know these differences. It has the sugar ribose, it's single-stranded, and it has uracil instead of thymine. There are 3 different types of RNA that you want to basically know. And what makes it really easy is that the name of the RNA basically tells us what it does. So the first one we talk about is rRNA, or we call that ribosomal RNA. And that basically makes up our ribosomes, so it combines with proteins to do that. The next one we have is tRNA, or transfer RNA. So this basically transfers amino acids to the mRNA codon during protein synthesis. So if you remember, amino acids are the monomers that make up the proteins. And then finally, our last one is the mRNA. So that is our messenger RNA. And so it basically carries the DNA message outside of the nucleus to basically start creating the code, or it carries the code to the transfer RNA. So we're going to go ahead and now start our 13.2, and we start 13.2 by talking about protein synthesis. And the first part of protein synthesis is going to be transcription. So this is where we have to rewrite the DNA message into RNA. So in transcription, segments of DNA serve as a template to produce complementary RNA molecules. So again, transcription, you can think we're rewriting, we're transcribing things. We can also, and you know what, I'll come back to that point later. So transcription, you're rewriting the DNA message. So to transcribe something is to basically write something down. Okay, so this is also a picture of transcription. You can see that the DNA is being unwound, and we're adding these RNA nucleotides to that DNA strand, or the complementary DNA strands. And the enzyme that does it is RNA polymerase. So we've heard of DNA polymerase, where we go with replication and new DNA strands are made. Now we're hearing about RNA polymerase. So when you hear RNA polymerase, you're making RNA. DNA polymerase, you're making DNA. So when this is done, RNAs require editing before we actually use them. So we have exons and introns. Basically what happens is introns are removed from the code, and exons are what is actually expressed. Now, why waste it? Why spend energy making a large RNA and just throw parts of it away? A lot of times it just allows us to express different genes, so depending on what you're trying to make, removing parts of the DNA will allow us to express different things. It may also have a role in evolution, and it allows small changes in genes to have basically a bigger effect. So basically, our master plan stays safe in the nucleus. So our DNA stays safe, and we want it to stay safe because that's the only plan your cell has. And so that's why we had to make our copies. So when we think of transcription, we're going from DNA to RNA, and that takes place in the nucleus. And if you look at that, then we go from to translation. So translation is where we translate that message. So we go from RNA into proteins, and we go to the proteins because the proteins are basically what does everything within our body. So the DNA is basically holds the instructions that tell the proteins what to do. So how can just 4 bases give directions to make 20 amino acids? So if you remember, amino acids are what make up our proteins. Basically, our message is read in groups of 3, and so we call those groups of 3 codons. So we have this top message up here, and this is, you can tell, an RNA message. Let's say it's the messenger RNA, and you can tell it's RNA because there's U's in it, okay, and we don't have any T's in them. So basically, if we were going to go to read this message, we would want to break it up into 3s, okay, in groups of 3s. And so you can see that we've broken it up down here, and each group is going to equal a different amino acid. So UCG equals serine, CAC is histidine, and GGU is glycine. And these codons, like I said, represent our different amino acids. We actually have 64 possible codons, and this is the little chart that you would use to be able to read what codon represents what amino acid. So for instance, if we look at the A, U, G, we see that that's methionine, and that's actually the start codon. So we always start with AUG. We have 3 different codons for our stop codon, and you can see we have the stop one out here, so that's UGA, and that's going to tell basically our ribosome to stop making that protein. And then we also have, let's see, where is it? Here's another stop. We have UAA or UAG. Now we're going to go ahead and look at our tRNA. So each tRNA has an anticodon, and that's basically what's connected to the bottom of the tRNA. It's the UAC here, and it's always going to match up with the codon, which is on the mRNA. So the codon on the mRNA is AUG. And remember, when you're trying to figure out what amino acid goes with that specific tRNA, you read the codon. You read what's on the mRNA sequence. So, like I said earlier, is that each tRNA carries only one kind of amino acid. So the AUG codon carries the tRNA that has the anticodon for that will carry the amino acid methionine. That's start amino acid. So the first step of protein synthesis is transcribing the DNA. So step 1, which is said right here in A, or which is stated here, we have to make the messenger RNA. And as we did in class, we went ahead and we used DNA polymerase to basically separate the DNA strands and then make the mRNA strands. So after that, we take that mRNA outside of the nucleus and into the cytoplasm, so translation can begin. Remember, we have the C before L, so transcription comes before translation. So there's a C in transcription and L in translation. So after we do that, our transfer RNA, or our tRNA, is going to match up, or the anticodon is going to match up with the codon, and it's going to go to the ribosome, and basically the ribosome is going to help bring the amino acids together. So you can kind of see how these tRNAs are found in the ribosome, and the ribosome is going to then attach those amino acids with a polypeptide bond. Okay, so you can see that. Once the tRNA is not used anymore and the amino acid is attached, we go ahead and it is released into the cytoplasm. And so the ribosome basically works along that mRNA strand like a little machine until it comes to that stop codon, where it tells the stop codon on the mRNA, where it tells the ribosome to basically stop making that polypeptide chain, aka that protein. So let's go ahead and just kind of summarize this. So DNA to DNA we call replication. And remember, replication happens via DNA polymerase. DNA to RNA we call transcription, and that happens via RNA polymerase. RNA to protein we call translation. So basically we translate that message into a protein. Okay, so this next part, I want you guys to go ahead and make sure that you get this into your notebook. So this is what I'm going to check tomorrow, making sure that you have this concept map. It basically summarizes R and RNA, the different types of it. So we have RNA at the top, and then we have all of our different types of RNA. And I want you to fill out the rest of these boxes, and we'll go through them in class. All right, guys, so